listeners, and welcome to episode 116 of Cell and Gene, the podcast. I'm Erin Harris, and you're about to hear my conversation with Dr. Arun Upadhyay, Chief Scientific Officer, Head of Research and Development at Ocugen, about a truly ambitious and game-changing chapter in gene therapy. Ocugen's focus is the retina, and they're tackling conditions like retinitis pigmentosa, Stargardt disease, and age-related retinal degeneration, but not in the way you might expect from traditional single-gene approaches. Arun walks us through how Ocugen's modifier gene platform works. By using a single gene therapy that resets disrupted networks in the retina, offering the potential to treat broad patient populations regardless of genetic mutation. You'll hear about their lead program, OCU400, currently in phase 3, and the scientific, clinical, and manufacturing lessons they've learned along the way. We'll also cover what's next for gene therapy in ophthalmology, how evolving endpoints, platform manufacturing strategies, and a better understanding of disease mechanisms are helping to accelerate progress toward restoring vision for patients with inherited retinal disorders. If you care about how gene-agnostic strategies, scalable manufacturing, and smart clinical design can move from rare retinal disease into broader realities of cell and gene therapy, then definitely stay tuned. And so here's my conversation with Ocugen's Dr. Arun Upadhyay. Enjoy. Hi, Arun. Welcome to Cell and Gene, the podcast. Thanks for your time.
Thank you for having me here for this podcast. Looking forward to speaking with you.
Yeah, same here. So, okay, tell us a little bit about Ocugen and the company's mission.
So Ocugen was founded in 2013, by Dr. Shankar Musundri and Dr. Kampela, with a mission to bring the game-changing, therapy to the, specifically, targeting ophthalmic indication. So at Ocugen, we are dedicated to bringing game-changing gene therapy to market and working even harder to provide access to patients globally. So currently we have programs in development for inherited retinal conditions like retinitis pigmentosa, Stargardt disease, and also the age-related, retinal conditions such as geographic atrophy. And these programs, most of these programs are in clinical development. Some are already in phase 3, phase 2, 3, and most all these programs are based on our breakthrough modifier gene therapy platform, which has potential to address significant unmet medical need for large patient population through our gene-agnostic approach. So based on the unique mechanism of action of these modifier gene therapy, they target diseases, in a gene-agnostic way or multifactorial way. And this means that they are not just going to target a small subset of patient population affected by single gene mutation, but conditions like retinitis pigmentosa, where we have hundreds of genes mutated, it has potential to tackle those all gene mutations, together. So that's where we are, and that's what. So our mission is basically to bring this modifier gene therapy technology and offer to the patients which are affected by various retinal diseases.
Great, thank you. And you, of course, mentioned your modifier gene therapy program platform, and the work you're doing for retinitis pigmentosa et al. And so that's what I want to talk about next. So the modifier gene platform, the gene therapy platform, you're advancing 3 programs toward BLA filings in the next 3 years. So I'd like you to really talk us through what they are and the scientific or technical advances that enabled the broad gene-agnostic clinical indications, particularly for complex areas like retinitis pigmentosa.
Very good question. So I will start with giving some background about the disease itself, and because that will probably help understanding, listener,
Sure.
that why we took that approach. So if you look at conditions like retinitis pigmentosa, uh, The disease is caused by this disease is like highly heterogeneous and complex in nature. Okay, and this complexity is attributed by not just because of the genetic heterogeneity of the disease itself, but there is a lot of clinical heterogeneity. And what does it mean? That depending on the nature of mutation and the genetic background of individual itself, the onset of the disease, progression of the disease, and the severity of the clinical symptoms, they all could be very varied. So there is a wide range of the clinical symptoms in these patients also. But one common thing with this particular disease is that it lead to the degeneration of photoreceptor. Okay, and that is the major driver why patients start losing their vision. So in nutshell, irrespective of what genetic background is, genetic cause is, or other attributing factors leading to the clinical symptoms, eventually they all drive or merge to the one single pathway, which lead to the degeneration of photoreceptor and subsequently loss of the vision. So when we want to tackle this kind of condition using gene augmentation approach or gene editing approach, where you are just targeting a single gene, you may be able to correct the defective gene, but as the disease has already advanced to the certain stage, there is no way those correction will be able to address the other downstream pathway which are already altered by that causative gene. Okay, and what does it mean? That already it has led to the what you call initiation of the cascade pathway, which are uh, basically leading to the photoreceptor death. Okay, so the way when we started this work, our focus was how to preserve the photoreceptor. Is there something can be done to preserve the photoreceptor irrespective of what genetic background is? And that is where modifier gene therapy comes into the picture. So what we learned in our preclinical research that in the animal model of various genetic background of the retinitis pigmentosa, one of the common pathway where we noticed that all these models were showing some biomarker was down regulation of the key master regulator genes like NR2E3, like our OCU400, or NR1D1, RORA, NRL, CRX. So these biomarkers, irrespective of what genetic mutation was, they were significantly down regulated in this mice model. And when you delivered this NR2E3 gene, that is OCU400, to these mice, it was able to bring back all those down-regulated genes to the normal physiological level. And this is what we call like molecular resetting. So when you do the molecular reset by delivering this master regulator gene NR2E3 in the retina of those mice model, you are able to do the molecular reset, and that subsequently lead to the bringing homeostasis in terms of cellular and functional pathway. And by doing so, we are able to preserve the photoreceptors and subsequently preserving the vision and halting the progression of the disease. So that is what in nutshell our modifier gene therapy is, and that's about NR2E3. Similarly, like other molecule like RORA, it is also a master regulator, and it targets multiple pathway in a disease like a Stargardt and macular degeneration, for example, geographic atrophy. So rather than, if you look at most of the current therapeutic, are either approved or in development, they target like, single pathway, largely like complement driven approach, complement targeting approach. Here we are looking at targeting multiple pathways which are associated with the disease pathophysiology, like oxidative stress, formation of drusen like lipofuscin like material because of altered lipid metabolism, complement pathway and inflammatory pathway. So that is how this modifier gene therapy approach is different and have potential to address the retinal diseases like RP and Stargardt in gene agnostic way, and at the same time address multifactorial condition like geographic atrophy and age-related macular degeneration by targeting multiple pathway linked with the disease pathophysiology.
Thank you. And I appreciate the background information leading up to where we are today. That's really helpful for our audience. Now, I want to talk a little bit about the phase 3 trials for OCU400 or OCU400, the first gene therapy trial to receive a broad FDA indication for retinitis pigmentosa for our audience members. What were the pivotal challenges in translating this modifier gene therapy from preclinical science to a late stage gene agnostic trial? What were those challenges, and how did your team overcome them, continue to overcome them? What's the status there?
Yeah, very good question, actually. So if you look at, like, suppose if you are developing something very big, to start with, of course, like before getting into the clinic, first in human trial, getting into clinic, we have to ensure that, yes, this molecule has that gene agnostic potential. Okay. So in the preclinical stage itself, our approach was to understand how these modifier gene, provide benefit in the diverse animal model of retinitis pigmentosa. So that was the first challenge. We have to kind of ensure that, we get the right kind of animal model representing different pathophysiology within the retinitis pigmentosa in terms of, the molecular pathway. For example, taking a rhodopsin gene mutation, which is more like, protein involved in phototransduction pathway, then taking like PD6 beta, which is more like enzymatic pathway in the phototransduction cascade. Then taking an example of some ciliary protein like CEP290 gene mutation, RD7 mice. So we try to include diverse genetic background, mice in preclinical research itself, and to see whether this molecule is able to provide benefit both in terms of preserving the photoreceptor as well as the functional improvement in these models or not. So that was the first thing we have to kind of demonstrate. So we were able to kind of, get those signals in the preclinical research. And that was the basis for us to take this into the human study. But one of the challenge in the human trial also, because these gene therapy trials, especially the phase 1, 2, these are not a big trial. You are limited by 3+3 design. Normally you do try like 12 to 20 patients, like so. So you cannot basically accommodate all the mutations. There is no way to represent every mutation in the early phase trial. So our approach was to, select the mutations which can be potentially used to demonstrate gene agnostic mechanism of this molecule in phase 1, 2 study. And that's why we started with the most common and no aggressive mutation, autosomal dominant rhodopsin gene mutation in our phase 1, 2 study. On top of it, we also chose the patients with, other mutations like NR2E3 autosomal dominant and autosomal recessive. So that is how we started our phase 1, 2 study. And when we looked at the phase 1, 2 study, data, what we noticed that, yes, indeed, whatever we saw in the preclinical work is getting translated into the human early phase trial in a gene agnostic way. And that led us, like, to move forward with our phase 3 study design, engage with FDA and get alignment on broader gene agnostic trial, which is we are doing right now. So our current trial is targeting broader RP patient population, irrespective of genetic background of individual, whether they are syndromic, non-syndromic. So we are basically encompassing all the RP patient population in ongoing pivotal study trial. But one of the learning we had and challenge which we noticed in our phase 1, 2 study is that because of the very, like, rare nature of this disease and heterogeneity, what I mentioned to you, clinical symptom heterogeneity, it is very difficult to identify, a endpoint, primary endpoint, which can be used for this diverse set of, patient population. So how we tackle that we took the learning from our phase 1, 2 study, and we worked on optimizing our primary endpoint, which is based on, approvable and acceptable endpoint from FDA, that is mobility course. But we further, optimized and optimize this endpoint in such a way that it can be utilized for the wider range of the patient population. And when I say wider range, means the patients with the early stage of RP to mid stage RP to advanced stage RP, because a lot of early stage RP patient, they show the ceiling effect in the prior mobility courses. So we have to modify the course in such a way that we can enroll early stage RP patient and still we can demonstrate the clinical benefit in those patients, through kind of, after delivering the OCU400, to those patients. So those are some of the critical challenges we faced, and of course, and another key area where we focused on was streamlining the platform, because you know that all our gene therapy pipeline is based on modifier genes. So in the beginning itself, we took a platform approach. So we are sticking with AAV5 vector for all our programs, and we are optimizing the condition in various stage of the development, from manufacturing and assay perspective, in such a way that it can be used as a platform for other pipeline, when we are diversifying these gene therapy to the other indications.
Great, thank you. And that leads us right very nicely into my next question for you. Kind of lessons for other biotechs who have diverse pipelines, knowing that with commercialization, of course, the therapy being delivered to the patient, with commercialization as a goal, but manufacturing and process validation being major hurdles. How have you and your team approached those two things when it comes to regulatory compliance and scalability? What kind of lessons learned could you share with other biotechs in the same space?
Sure. So I think when we started this program, we were pretty much, there are few things which were certain about that. We are targeting, like, say, rare indication. Okay, so we had a fairly good understanding about what a skill we may be kind of looking for, for commercialization perspective. Okay. So when we were developing the process for our very first, lead candidate, OCU400, our goal was, to optimize most of the process parameter, in such a way that it is scalable. And not only that, we also wanted to ensure that we basically do the manufacturing pretty much at close to the commercial scale in our early phase clinical study itself. Okay, so that's the approach we took. And also from CMC perspective, one of the key aspect is for gene therapy having like control on your a strength determining assay, that is VG titer, and good understanding around the potency assay. So we started working on these two in the very early stage of development, even when we were in preclinical stage, to optimize this, the VG titer assay, and implement in the early GMP manufacturing, and also starting, developing a potency assay, which are demonstrative of the biological function of your transgene in the cell culture model. That was our approach. And in parallel, also we took matrix approach, where like multiple orthogonal assay can be utilized to demonstrate the overall potency of the molecule. Regarding another area, we focused that we know that we are targeting the retinal condition. It is local delivery. It is not systemic. Okay, and our focus was to achieve the highest purity for the full capsid. So one area we spent quite a bit of time in optimizing process was targeting to achieve the capsid purity more than 80%. Okay, full capsid in the product. So we spent quite a bit time, optimizing downstream condition and reaching, and identifying the parameter which can lead to that purity. So by, having implementing the early process controls and in-process monitoring, and having the analytics ready to monitor those, signals which are indicative of purity and capsid enrichment, and also optimizing the condition to lead to the, optimized critical quality attributes of the product. So all these parameters were considered during phase 1-2 stage itself. So when we are advancing this program to, like, say, later stage in commercialization, there's a very minimal, a scale up work we had to do, and that is how we have been able to tackle. So our process validation is more like, pretty much based on whatever we did for our phase 3 study when we transitioned from phase 1 to phase 3. So we did like one scale up, and right now basically we are doing the process validation run and following those parameters which were already locked during like phase 3 study.
Excellent. Thank you. I want to talk a little bit more broadly about the major clinical challenge that still impact gene therapies for inherited retinal diseases and blindness more broadly. So, what's still challenging us, and also what are the promises? Not necessarily promises, but what's the optimism for the future for progress?
So one of the challenge I think primarily we have been facing in this field is of course attributed to a lot of failures, am I right? Not able to demonstrate. I'm talking particularly in about like our space like inherited retinal condition has been demonstrating the clinical benefit on the meaning, you can say like meaningful clinical benefit in these patients. So some of the past trial our company you can say failing primarily on those aspects. uh, and another challenge is of course most of the people are taking approach which are either gene augmentation or gene editing, so targeting a small patient population. So those genetic correction may be able to fix the defective gene, but as I highlighted in the beginning, they may not have a full potential to uh, prevent the progression of the degeneration of the photoreceptors. And if we cannot achieve that, then it is very difficult to kind of provide the meaningful benefit to these patients. So some of the approaches which I think currently we see in the field, which are more towards either finding a factor or approach which can prevent the progression or halt the degeneration of the photoreceptors. And other approaches also we see like optogenetics and other modality where they are trying to kind of see if in the patients which are very advanced a stage of the disease, how this optogenetic approach can deliver a factor which can help in like bringing some vision to the advanced stage of the RP patients. But besides that, another challenge which I think in general we faced in this field was uh, looking for the right primary endpoint, because this IRD condition is very slow progressing disease. And so when we look at like traditional endpoints like looking at the visual acuity, it doesn't change a lot over course of 1 year or 2 year. So if we really want to kind of demonstrate the clinical meaningfulness on those parameters, then running the trial for like 4 or 5 years to get the signal is not an easy task, and not like everyone is invested in that. So, 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 so one area I think in last 4 or 5 year field has evolved is looking for the new, primary endpoint, which can be, meaningful as well as measurable within the short period of time. And one of those, endpoint has been like, say, for example, mobility course. Now in the field, we are talking about like, EG area, or low luminescence visual acuity. So these are some of the development I see, and which I believe that will be able to address some of the challenges we had in the past in terms of, bringing the therapy to the market.
Good. Thank you.
Mhm.
Finally, before I let you go, one more question. With the industry attention on scaling new gene therapies and securing regulatory approvals, where do you see the most exciting opportunities for cell and gene therapies to address wider unmet needs in ophthalmology?
So in our space, of course, in ophthalmic space, the right now focus is still I see a lot of company, still interested in targeting a single gene mutation. But now we see more and more discussion around coming up with the approaches which has ability to address the wider set of genetic mutation in these conditions. So gene agnostic approach, I do see that when we started talking about modifier gene therapy 5 years back, probably we were the only one, I think, at that time when we started the trial. but now I see that there are many who are talking about gene-agnostic approach, either you know uh, through delivering the neurotropic factor, which are neuroprotective in nature, or you can say optogenetic approaches. But one of the advantage I see of modified gene therapy approach is that if this platform is proven in this retinal condition, this can be applied across other disease area also. For example, age-related condition, dementia, Alzheimer, for example, or other conditions which are, neurodevelopmental in nature, like autism spectrum disorder, or the diseases which are like multifactorial in nature. So I see that, that gene therapy approaches like which have ability to target holistic pathway could be the next, next revolution in gene therapy approach. So going away from just targeting, single gene, single mutation, correcting it, to using gene therapy as a tool to deliver a factor which can have a broader, uh, impact on the cellular and molecular pathway, and thereby addressing the complex diseases.
Excellent. Thank you. All right, listeners, that is a wrap on this episode feature Dr. Arun Upadhyay. Arun, thank you so much for your time. This was very valuable and your insight was exceptional. Thank you for your time.
Thank you. Thanks, Erin. Thanks for having me.
Absolutely. I enjoyed our talk. Thanks, listeners, for tuning in. Please subscribe to Cell and Gene, the podcast, so you never miss a valuable episode just like this one. And we'll talk to you soon.